Hello, everybody. We'll continue our discussion on to the clinical microbiology, and today we will take up a very, very important topic that has almost affected one-third population of the entire world already. The incidence of these infections, there are every year about 1.5 million cases, and the organism, it causes the death of more than 1 million people every year in the entire world. The organism has been adopted to the multi-drug resistance pattern, and there are almost 0.5 million cases each year which are affected by the multiple drug resistance microorganism of this bacteria. Now, the disease has been, you see, on high trend because of the presence of the HIV, and this is what we called as the awakening of a sleeping giant. The disease was sleeping, but it has been re-emerged because of the infections of the HIV. And the WHO says that the tuberculosis is the world deadliest infectious disease. And in the recent pandemic or epidemic in the corona or this COVID-19, this coronavirus death tolls, it has mounted to a lot. And the global health authorities, they are reminding that the tuberculosis is the deadliest infectious disease in the entire world. So in the current scenario of COVID-19, and its pandemic state and environment of high mortality and morbidity, we should not forget about another disease which is really causing a huge mortality in the world, and the disease is the tuberculosis. And we have over 1 million deaths reported each year because of this infection. So highly infectious disease that causes huge mortality and morbidity is the tuberculosis. Now, the causative organism of this microorganism of this genus is the genus name is Mycobacterium, and the causative organism is the Mycobacterium tuberculosis. We have many species of the Mycobacterium tuberculosis, but before we move on to the species, let's have the characteristics of the genus of the Mycobacteria. We all know that mycobacteria, they are the acid-fast microorganism. They are not gram-positive, they are not gram-negative, but they have a different, what you call as the cell wall structure, and that cell wall structure gave it specific staining characteristics, and this is stained with the help of a gram stain procedure, or what we call as the Zn stain. The organism can be hardly stained with that of the gram stain procedure. Now, the cell wall of a gram-positive, the Mycobacterium tuberculosis, it has a unique waxy material inside the cell wall. And this waxy material contributes towards its virulence along with that of its staining characteristics. We can see over here the red cells, red color cells, this is the acid-fast stains, long rods of Mycobacterium tuberculosis that can be seen against the background of the blue color. The organism is obligatory aerobic, and this makes sense that why this microorganism is really proliferative in the lungs. The lung is the most oxygenated area, particularly the upper part of the lungs. So you will find the primary lesions in the lungs, and that makes sense because of the characteristics of this microorganism. It is obligatory aerobe, and it prefers to live in an area of the body where there is oxygen supply is huge, like the lungs and like the kidneys. Now, the cell wall of this organism, are the members of this genus Mycobacteria, they have high concentration of lipids. About 60% of the cell wall is made up of lipids. It does not have peptidoglycan, does have, but very, very small component of the peptidoglycan compared to that of the gram-positive cell wall. The colonies that it forms onto the medium, it is a typical granular waxy pattern of growth. And if you look at this, this is the medium for the growth of Mycobacterium tuberculosis, as you already know. It is the Lensen-Janssen medium. This medium is rich in egg and the other components. The LJ medium is the preferred medium for the growth of this microorganism, and you will see the granular waxy colonies that appear onto the green color medium of the LJ medium. LJ medium contains, you see, the egg yolk along with that of the malachite green, which is a dye, and this dye is used, just to inhibit the growth of the other contaminants that are present in the respiratory tract, and it will just allow the Mycobacterium tuberculosis to grow, but it will inhibit the growth of the other organisms which are present in the respiratory tract. Because this organism, the Mycobacterium tuberculosis, it is, you see, resistant to many of the chemicals, and it is resistant to acid and alkalis. That is why the sodium hydrochloride, we treat the sputum. When we take the sputum from the sample, from the lungs of a mycobacterial affected or infected possible candidate, what we do, we take the sputum sample and we treat the sputum with the help of sodium hydroxide. This sodium hydroxide, it will kill the contaminants or the other normal flora or the other organism which may be present in the sputum, but it will not kill the Mycobacterium tuberculosis. So we can really then make a smear, stain it with the acid phosphate, see it on the microscope, and you will have the broad red colors, bacilli of acid phosphate nature, because of the background is a blue in color. The multi-drug resistance Mycobacterium tuberculosis, they are becoming more and more. The incidence of MDR Mycobacterium tuberculosis, they are resistant day by day. And this resistance is mediated at the chromosomal level because the organism, it does not have any plasmids. So as we know that the the antibiotic therapy or the antimicrobial drug resistance, the genes are being present either onto the plasmid in most of the cases, and we have seen in most of the cases of the gram-negative rods. But in this case, the antimicrobial drug resistance is chromosomally mediated because this organism, it does not have plasmids. It does not have a plasmid. It's a plasmid-free microorganism, but it does have a chromosome, and the drug-resistant genes are located onto the chromosome. So, the acid phosphate microorganism, high contents of lipids, granular colonies on the algae medium, highly resistant to the acid and alkalis, and the MDR stains have their resistant genes present on the chromosome of the organism. Now, the mycobacteria broadly, you see, we have more than 50 species of the mycobacteria. But the major pathogens we have can be divided into three typical categories. We have the typical mycobacteria. We have the atypical or microbacteria other than tubercle bacilli, what you call the MART. This is also known as the non-tuberculous mycobacteria. So we have a typical bacteria, mycobacteria, we have atypical mycobacteria, and we have rapid growing mycobacteria. So three different names given to the mycobacteria. Typical mycobacteria are the Mycobacterium tuberculosis, Mycobacterium bovis, and Mycobacterium leprae. This causes the tuberculosis. Mycobacterium tuberculosis causes tuberculosis in humans. The humans are the natural main reservoir. Mycobacterium bovis is mostly found in the cattle and bovines, in buffaloes and cattle. And they transmit the disease with the help of the milk, because the milk given of these animals, the bovines, cattle and buffaloes, if it is contaminated, it is infected with the Mycobacterium tuberculosis, they carry this infection. They cause what you call as the gastrointestinal tuberculosis. And Mycobacterium leprae, you see, it is another organism which causes very, very chronic granulomatous disease, what you call as the leprosy. So these two are the causes of tuberculosis, one in humans, other one in bovines, but the disease from bovines to human is transmissible because of if you take the unpasteurized milk, there is every possibility that you can get the organism. Maybe you will not get the infection, will not have the disease, but still you are the carrier of the Mycobacterium bovis. Atypical mycobacteria are the mycobacteria other than tuberculosis are the non-tuberculous mycobacteria. They cause tuberculosis-like disease, not the real tuberculosis, but the tuberculosis disease, particularly in the AIDS individual. AIDS individual, the Mycobacterium avium complex, what we call as the MSC-MAC. Mycobacterium avium intracellular complex is the infection that we see mostly in the immunocompromised individual like AIDS individual. And we have another organism, what we call as the Mycobacterium kansasi. Now, the rapid growing microorganism, the mycobacteria, the Mycobacterium colony is the rapid growing mycobacteria. It causes rare human diseases in immunocompromised individual and an individual who has been implanted with the catheter or the devices. So typical mycobacteria, Mycobacterium tuberculosis and bovis causes tuberculosis, leprosy causes leprosy. Atypical or slow-growing microorganism, again the slow-growing microorganism, what we call the NTM or the MOT organism. This one is the most common, avian complex, and the rapid growing is the clony, which causes human infections, particularly in the immunocompromised individual, an individual who has been implanted with the foreign devices. Now, the Mycobacterium tuberculosis, what we are called as the acid-fast bacilli. The outer envelope, it contains a variety of high molecular weight complex lipids. This is very characteristic. The high contents of mycolic acid and waxy material is present in the envelope of the mycobacteria. And they have great role to play in the pathogenesis of this infection and the pathogenesis of the infection because of the mycobacterium. We have to go for the alternate staining characteristic because of the change in the cell wall components. And this cell wall is considered to be highly resistant. The cell wall has a profound adjuvant activity. This cell wall, because of the presence of various components that we will discuss right now in the next slides, it promotes the immunogenic responsiveness of the host, of the infected individual. Though huge diversity, a large number of proteins molecules are carried out by the cell envelope, cell membrane, along with that of the cell wall, and that combination of various proteins, they promote the immunogenic response of the individual. That's why we get the very early response of the cell-mediated immune type. The typical mycobacteria, as I said earlier, is the leading cause of death from single infectious diseases. Almost 1/3 population is infected. 1.7 million death that has been reduced now to the recent survey that I saw onto the CDC and the WHO site. It is now, see, almost close 1.3 million deaths every year compared to that of the 1.57 million that has been written onto the book that we use for this purpose. 9 million new cases every year is estimated, 0.5 million infected with the multi-drug resistant Mycobacterium tuberculosis. So this tells us that we should not go for the monotherapy against the Mycobacterium tuberculosis, because the monotherapy provides best opportunity to the bacteria like Mycobacterium tuberculosis to become resistant. So we always go for the multiple therapy. Now, even the Mycobacterium tuberculosis, which are resistant to the multiple drug organism has been resistance. So these incidents, they are increasing. The MDR, Mycobacterium tuberculosis, are increasing because of the AIDS. So AIDS, coupled with that of the Mycobacterium tuberculosis, very difficult to treat the individual. The high frequency of the tuberculosis, it is mostly found into the poor nutrition, overcrowding, lack of control measures, and this is the disease what we call as the poor socioeconomical population of the world. The organism does not produce any toxin, no endotoxins, no enzyme that contribute toward the infectiousness, but they do have some of the specialized protein that contribute towards the virulence, or they act as the virulence determinants of this microorganism. The distinguishing features, they are slow growing, particularly onto the media that we use. They can be grown onto the laboratory media, except the Mycobacterium leprae. Mycobacterium leprae is an intercellular organism that cannot be grown onto the ordinary media. It requires the living cell for its growth. The generation time for the Mycobacterium tuberculosis and other slow-growing microorganisms almost 18 hours compared to that of almost 20 minutes or less than 1 hour in many of the other microorganisms. the obligate aerobic microorganism, and the acid fastness of this organism, that why this organism is resistant to the action of the acid in the gram stain and then the acid fast staining procedure, is because of the mycolic acid which is present in the cell wall of the Mycobacterium tuberculosis. Now we'll have to take another two structures which are present in the cell of Mycobacterium tuberculosis. It has a cord factor. It has a cord factor. We will learn many new terminologies and very few of the new names for the lineage, and of course the component or virulence determinant that contribute toward the virulence of this organism that we have not studied before. It has a cord factor which is you see its chemical, it is the trehalose dimycolate, and it's correlate with the virulence of the stains, and it provides what we call it the serpentine cord-like growth. We will see in the next slide that how this growth look like. I'm sure that we have seen the slide of Mycobacterium tuberculosis, and we have shown you the cord factor that gives the serpentine-like or like growth of the microorganism. Now, the cell wall lipid is another the component, which is again chemically, it's a very different, and it requires for the pathogenesis in the lungs. So the cell wall components, what we call that the thiocyrrole dimycocerosate, it is required for the pathogenesis in the lung. So it has a cord factor, and it has a cell wall lipids component. It has a rich mycolic acid layer, and they all contribute towards the virulence of this microorganism. The organism can survive in the dried expectorated sputum, and that provides the basis for the aerosol transmission of this microorganism. The organism can survive even in the dry sputum, and once the sputum dries, it turns into particles, and these particles can be suspended as a dry air in the environment. So this is what we call as in the dry air and the wet air also. So this is what we call as the environmental transmission with reference to particular emphasis on the aerosol transmission of this microorganism. Mycobacterium tuberculosis, how do you see, look at this slide, how does the cord-like formation in the infected tissues look like? This is red stained strands, they're elongated, and they have the masked filaments of tubercle bacilli, and this is because of the cord factor. So the most stains, they contain the complex host and the cord factor. The cell wall antigens, which are present in the cell membrane and in the cell wall, they protect from the being destroyed by the lysosome of macrophages. We'll go into the pathology or the pathogenesis and let you know that how the cell wall antigen, they protect it from the macrophages and the action of the lysosome of the macrophages. And they contribute, the cord factors, they contribute towards the invasion and what we call as the persistence as intracellular parasite of the microorganism. We will deal on its process, that how this cord factor, it prevents the lysosomal action of the macrophages, and how does it provide opportunity for the invasion of the intracellular survival of this microorganism. Now, the mode of transmission of this organism is, as we all know, that is exclusively from the fine respiratory aerosol or droplet of the mucus, and particularly by coughing of the smear positive. Now, smear positive organism individuals are those patients that when we make when we take a sputum, we treat it with the potassium hydroxide, and then we make a smear and stain it on the microscope and see that we find few number of microorganisms. An adequate number of the microorganism in the slide, we call it a smear positive. So smear positive individuals, they are the main source of spread of infections. And the organism is very resistant and can survive for months. The pronounced effect is among the people sharing close, small rooms and limited access to air. This is what we call as the socioeconomically disadvantaged people, who are poor, who cannot have the good nutrition, who cannot have the access to the open air. They're living in the closed housing compartments. Humans are the natural reservoir, And another stain, this Mycobacterium bovis, it is present in the milk from the infected bovine, and if we do not pasteurize this milk, this means the unpasteurized milk from the infected bovine are the main source. But if you pasteurize the milk, or if you boil the milk, there are least chances that you will get the transfer of this microorganism. Before we embark onto the pathogenesis of the Mycobacterium tuberculosis, it is important to understand the virulence determinants. And as we all know that the Mycobacterium tuberculosis, it does not have any enzyme or toxins, maybe endo or isotoxins. So what happens, you see, the organism has some specialized component in its structure that act as virulence determinants. Now, if we see that the most important component that is present in the cell envelope and that act as virulence determinant is the sulfatides. So the sulfatides, along with that of another chemical substance which is released by the bacteria, is the what we call as the exported repetitive protein. This is abbreviated as, we all know it's abbreviated as EPR. Now what happens, once the organism, it enters into the body and it infects in the alveolar macrophages along with that of the other reticular endothelial cells, and the organism survives and multiplies within the cellular vacuoles, what we call as the phagosome. Now the phagosome and lysosomal fusion, that causes the destruction of the bacterial cell. But the component, the sulfatides, along with that of the, what we call as the exported repetitive proteins, they inhibit the phagosome lysosome fusion. Therefore, the action of the lysozyme to kill or to destroy the microorganism is inhibited. And the organism is survived intracellularly. Now, if the fusion does occur and the organism has the, exposure to be destroyed by the enzyme, now the second mechanism of defense or the second virulence factor that is present in the bacterial cell envelope in the form of vaccinator, it comes into the vax, then come into action. And it tries to reduce the killing effect of the lysosome onto the bacteria. So dual mechanisms. First, it prevents the inhibition or it inhibits the phagosomal lysosomal fusion. And if does it occur, then the vaccine nature of the cell, it tries to reduce the killing effect. Now, apart from this, we have another factor, what we call as the cord factor. This cord factor, which is chemically a T-halose dimycolate, it is present in the cell wall of the bacteria, the Mycobacterium tuberculosis. So one determinant, it is present in the cell envelope, which is sulfatides. The other one is cord, which is present in the cell wall. And this cord factor, it inhibits the leukocytic migration, and therefore it stops the mitochondrial respiration and phosphorylation of the cell. And this is required for the pathogenesis in the lungs. Now, along with the sulfatides and the exported repetitive proteins and cord factors, is how you see the surface proteins, what we call the tuberculins. Now, this is very important. This component of the bacteria is very, very important. These surface proteins, along with the mycolic acid and waxes, they really enhances delayed the hypersensitivity reaction. They are responsible for the hypersensitivity reaction and the initiation of the cell-mediated immune response. Now, these proteins, the tuberculin protein, these are used for the detection of the tuberculous antibodies in the human serum. And these tuberculin, they form the basis for the tuberculin skin test for the detection or for the survey or for the surveillance of the Mycobacterium tuberculosis. This is what we call as the purified protein derivatives also. So these tuberculin, along with the mycolic acid and the vaccines, they really all contribute towards the virulence of the microorganism. There is a delayed hypersensitivity and leads to the granuloma formation, and then it turns into the caseation because of the cell-mediated immune response. There is no toxins, either exo or toxin, and the damage is mostly done by the immune system. So these proteins, a group of cerebral proteins, along with the mycolic acid, along with the sulfatides, and they all contribute towards the virulence of this microorganism. Now, with reference to the lesions, you see the microorganism, Mycobacterium tuberculosis, produces 2-3 different types of lesions. It produces exudative lesions, which are really the acute inflammatory response to the bodies, and it occurs mainly in the lungs because the organism, the main site of redirection is the lung, the upper portion of the lungs where this oxygen is in abundant, and at the initial site of the infection, it really have the exudative lesions. Now, it also makes the granulomatous lesions, and as we all know that the granulomatous lesions are because of the chronic response. There is central area of giant cells that contain the bacilli, and then it is surrounded by the epithelial cells, and this is what we call as the Langerhans cells. Now, these Langerhans cells, when we make a histopathology or histoslide of the lungs to detect the tuberculosis, Langerhans cells are the pathognomonic lesions that occurs in the lung. Now, apart from the exudative and the granulomatous lesions, the organism can enter into the tubercle formation form. This is granuloma. We have the initial lesions are the exudative lesions, can pass on to the granulomatous form, and granuloma can be surrounded by the fibrous tissues. And then this fibrous tissue undergo the central caseation, and it can later on be healed by the fibrosis and the calcification. This is what we're called as the tubercles. So we have the exudative lesions, we can have the granulomatous lesion, and we can have the tuberculous lesion. That all depends upon the immune response of the body and the organ or the area involved by the microorganism. Now, with reference to the pathogenesis, the tuberculosis can broadly be divided into the primary tuberculosis, or it may be secondary tuberculosis, it may be reactivated tuberculosis, and extrapulmonary tuberculosis. Now, if you look at the primary tuberculosis, the primary lesions of tuberculosis at the site of initial infection, they are known as Gons focus. The technical name for the primary lesions produced by Mycobacterium tuberculosis at the site of initial infection, it is known as the Gons focus. Now, they are the alveolar macrophages that infect the bacilli, and then they replicate and lyse and infect the other organism. Now, after the primary lesion, if you see there is replication and further spread, then it enters into another type of lesion, what we call as the Gons complex. Gons complex are normally the involvement of the adjacent and distant lymph nodes. So there is parenchymal exudative lungs lesions. Along with that are the infected and enlarged healer lymph node. This is what we call as the Gons complex. So we have the primary lesions known as Gons focus, and we have the later lesions that involve the lymph nodes, what we call as the Gons complex. Now let's stay here and see sometime there. What happens with the Gons comes? Once after the entrance of the organism, the bacilli, they establish the infection, and they form the Gons focus, which usually occur at the lower lobe. Now, there are 2-3 possibilities at this stage when there is a Gons focus. What can happen further? Either the lesions, the primary lesions, they can heal by fibrosis, or they can progress to the lung disease, particularly in the individual who are HIV positive, or who are malnourished, or they can lead into the severe bacteremia, and they can enter into the what we call as the hematogenous dissemination. Now, everything that has to be progressed, it has to be progressed through by the focus, by the Gons focus. Initially, the body tries to heal it, and it heals by fibrosis. and there is delayed hypersensitivity reaction. And this stage, the tuberculin test, which is used for the screening of the tubercle bacilli infected individuals, they are positive. So this tuberculin test is positive at Ghon's focus stage when it heals by fibrosis. Now, if it progresses, then it may lead to death. But the severe bacteremia can lead to another type of tuberculosis, which is scattered in the lung area, what we call as the miliary tuberculosis. And then the ultimate fate of this miliary tuberculosis, if not treated properly in time, is again the death of the individual. Now, what happens in case of lymphatic or hematogenous dissemination in this area? There is a dormant bacteria bacilli in several organs, and they later go into the latent tuberculosis. And then there is a reactivation can also occur from this latent tuberculosis. And there is other cavity lesions, usually in the upper lobe area. And ultimately, you see, from here, it can spread into the extrapulmonary tuberculosis, and there is involvement of the lymph nodes, the renal, GIT, kidneys, and vertebral body, and the central nervous system. So this is how the lesions of the primary pulmonary tuberculosis from Ghon's focus can spread to the other body area, can be healed, they can be disseminated through the lymphatics and blood, goes to the other body organs, or they can be develop severe bacteremia that result into biliary tuberculosis. So we'll take all of these one by one now. But if you look at this, you see the primary tuberculosis, Ghon's focus, either it will heal by fibrosis, there is delayed you see the hypersensitivity reaction, and there is a tubercular test is positive at this stage. Now, if it progressive lung disease would occur, particularly in the HIV positive individual, and there may be death. There may be severe bacteremia, which result into the spread of miliary foci, the tubercle foci, into the various other body, lung sites. But this is what we call as the miliary tuberculosis. And if not treated, along with that of the progressive lung disease, it can lead to the death of the individual. The lymphatic and the hematogenous dissemination is there. There is latent tuberculosis can develop. This latent tuberculosis can reactivate because of the compromising factors, and there is cavitary lesions and the death and the multiple organ involvement by Mycobacterium tuberculosis. The primary pulmonary tuberculosis, also infection, you see, individual encountered by the Mycobacterium tuberculosis for the first time. The bacilli, they are phagocytized by the alveolar macrophages. And if they're not phagocytized, they will survive and replicate intracellularly, and killing macrophages until the cell-mediated immune response is initiated and the Ghon's focus is formed. Now, the non-resident macrophages, they are detected at this site. They're ingest the mycobacteria and transport the wild lymphatics to the regional lymph nodes, what we call as the Ghon complex. So the primary lesions at the tissues are Ghon's focus. When these lesions, they involve the surrounding lymph nodes, they call it as Ghon's complex. And the most people, they heal without disease, and the organism, it was of the Ghon's complex and remains viable unless treated. Now, this is how the primary tuberculosis, there is large influx of the monoleukocyte. This is the histopathological slide that you can see in the tubercles. Mononuclear cells, large, results in the formation of specific infectious sites, what we call as the tubercles. Pathologically, tubercles are the granulomas. And this is very important. See, you may have question for the granulomatous structure of the Mycobacterium tuberculosis. Along with that, sometimes they say that describe or SQs type questions regarding the granuloma forming bacterial infections, tuberculosis, and syphilis. So if how the granuloma would look like, you see, the tubercle, pathologically it's a granuloma. It comprises of the central core of the tubercle bacilli, and the large macrophages, outer wall of the fibroblasts and lymphocytes and neutrophils. This is how this is called. This is what is the epithelial cells you can see over here. This is epithelial cells, and this is how the tubercle or the granuloma would look like. And this is histopathology. You can have, you see the caseous necrosis. This caseous necrosis all around. This is the granuloma cells, the giant cells, if you look at this. So this is how the histopathologically would look like, and this is how it really comes out of the granuloma. All right. The primary tuberculosis, tubercle, as we said earlier, they can heal at some stage of life and become fibrotic, or they frequently, you see the centers of tubercle, they break down into the necrotic caseous lesions, and they gradually heal by calcification and persist for a lifetime. Sorry. A reactivation takes a long time. The bacteria can be, bacilli can be dormant for weeks, months, and for many years and years. And the tubercle filled with the masses of bacilli, they explode, they expand, and they drain into the bronchial tube. And this drainage in the bronchial tube and the upper respiratory tract can lead to the dissemination of the disease. Now, reactivation tuberculosis, it normally rises or reactivate from the latent form, and consequence of impaired immune function, the impaired cell-mediated immune responses there that result in the formation of, from the malnutrition, infectious diseases, or chemotherapy. So whenever your body is under test, you become immunocompromised because of the chemotherapy, because of many other diseases, because of some infections or malnutrition, and then the reactivation would take place. And this is a potential infection, can affect any organ, may develop into a disseminated form, what we call as the miliary tuberculosis. So reactivation always is, you see, leads to the spread of the disease. The site other than the lungs may be involved, including the lymph nodes, kidney, long bones, genital tract, brain, and meningitis. And as a principle, it is generally said that tuberculous bacilli can infect any tissue of the body except hair and nail. So you can have tuberculosis anywhere in the body, any organ, any tissue, except the hair and the nails, which are almost, you see, the very tissue that you see, the nail are almost the dead tissues. So any organ, the tuberculosis, the lung TB, the genital tract, the brain, meninges, lungs, anything can be involved for that. So keep in mind, as a general principles, any organ of the body or any tissue can be affected. Now this is how the miliary tuberculosis will look like. If you say the cut surface of the lungs, which is covered with white nodules, if you look at this, is white nodules covered with white nodules. This is the miliary foci of the tuberculosis. A small percentage, of course, not much of these, the small percentage of the individuals who develop severe bacteremia and later on enter into the miliary tuberculosis, particularly in the immunocompromised. The bacilli, they are not contained in the tubercle, but they involve the blood streams and the cases dissemination, and the dissemination can also occur. This is how the tissue would look like if you make a histoslide of the affected part of the lungs. The clinical findings of this disease are, you see, 90% of the individuals, they are the carriers, they are asymptomatic. And we will discuss, when we will discuss the prevention, that how this mycobacterium tuberculosis infection, the tuberculosis, it mimicked with that of the coronavirus. And what was the initial response of the humans when this was in the early, back in 1935, 30s, were first time discovered. So 90% of the individuals, as we see in case of corona, we are asymptomatic, many, maybe we don't have the exact data, but I'm sure that all of us now have been exposed to the corona infection. There's low-grade fever, fatigue, the night sweating, weight loss, anorexia. These are general consideration. Then, of course, in the later stage, when the focus is developed, then there is violent coughing, and there is greenish or bloody sputum that depends upon the penetration of the tissue by the cells, the bacilli. Now, there may be, you see, a specialized lesion, what we call as the crufola. Crufola are the uni or bilateral clinical lymphadenitis. Now, lymph node TB is very common. This is one of the most important and most frequent manifestation of the mycobacterium tuberculosis. The lymph nodes, they are swollen, non-tender, and the cervical lymph node, but any lymph can be involved. From the cervical lymph nodes, it can disseminate to any area of the body. This is what we call this crufola. Then maybe you see the erythema nodosum. This is the tender nodules along the extensive surface of the tibia and ulna, and you can see the redness, the ulceratic area. There are some type of lesions present onto the surface of the tibia and ulna, and 40% of the mortality if you do not treat this kind of tuberculosis. This was all about the generalized clinical signs and symptoms. We will enter into the extrapulmonary tuberculosis of the various organs of the body next time. Thank you very much.